This module describing the process of urine formation is a part of the paper on human physiology. It describes the different processes and stages involved in urine formation. The learner will be able to define and comprehend the meaning of urine formation, explain the stages in the process of urine formation, and describe the actual ultrafiltration mechanism. The module aims to explain the processes involved in urine formation. Now, let us recall the location and external anatomy of the kidney. The kidneys are two bean-shaped organs located outside the peritoneum at the posterior portion of the upper abdomen. They are located one on each side of the vertebral column and are protected by the ribs and a layer of fat. The renal artery, renal vein, and ureter connect to the kidney at the indented media border called as the hilus. The renal hilus is concave and leads into a renal sinus where the blood vessels, nerves, and lymphatics lie. The kidneys are surrounded by a fibrous, transparent renal capsule, a fatty adipose capsule that cushions the organ, and an outer fibrous renal fascia that anchors the kidney to its surrounding structures. You can refresh your memory looking at the figure which depicts the location and external anatomy of the renal system. Now, let us move on to the process of urine formation. The process and mechanism related to urine formation involves complex functions, namely ultrafiltration, concentration, and secretion of substances which help to form and excrete urine, the most voluminous excretory product of the body. About 1 to 1.5 liters of urine is formed each day. The nephron is the structural and functional unit of the kidney, and it has two parts, namely the Malpighian corpuscle and the renal tubule. The renal or Malpighian corpuscle consists of two portions, the glomerulus and the Bowman's capsule, which encloses the glomerulus. The renal tubule has three parts, the proximal convoluted tubule, loop of Henle, and the distal convoluted tubule. The urinary apparatus thus consists of the glomerulus, Bowman's capsule, and the renal tubule, and involves the three vital processes of glomerular filtration, tubular reabsorption, and tubular secretion. Blood passes through the glomerular capillaries and the plasma is filtered into the Bowman's capsule. This is called glomerular filtration. The filtrate passes through the tubular part of the nephron and undergoes various changes both in quality and in quantity. Substances like glucose, amino acids, water, and electrolytes are reabsorbed from the tubules. This process is called tubular reabsorption. Finally, certain unwanted substances are secreted into the tubule from peritubular blood vessels. This is called tubular secretion or excretion. While filtration is the function of the glomerulus, reabsorption and secretion are the functions of the tubules of the nephron. Here you can see the diagrammatic version of the nephron. You can see the nephron, the different parts in terms of the functions they perform in different stages. Stage 1 glomerular filtration, stage 2 tubular reabsorption, and stage 3 tubular secretion. To repeat, the mechanism of urine formation consists of three distinct stages that are as follows: glomerular filtration, tubular reabsorption, and tubular secretion. Stage 1 comprises of glomerular filtration. It is the process in which water and other minute molecules such as electrolytes, glucose, fatty acids, amino acids, nitrogenous wastes, and vitamins in the blood plasma pass from the capillaries of the glomerulus into the Bowman's capsule. That is, all substances of the plasma except the plasma proteins are filtered, and they are in the same concentration in the glomerular filtrate as in the blood plasma. Some substances such as calcium, iron and thyroid hormone in the blood are bound to plasma proteins that retard their filtration by the kidneys and are retained in the bloodstream. The small fractions that are not bound to plasma protein, however, pass freely and appear in the urine. That is, the substances pass through a filtering membrane formed by three layers, namely the endothelium of glomerular capillary membrane, the basement membrane and the visceral epithelium of the Bowman's capsule. Glomerular filtration is also called ultrafiltration, since even the minute particles are filtered. It is thus a passive, non-selective process in which hydrostatic pressure forces fluids through the glomerular membrane. Next, we move on to what glomerular filtration rate is. Glomerular filtration rate is the total quantity of filtrate formed in all the nephrons of both the kidneys in a given unit of time. That is, it is the volume of filtrate formed by each minute by all the glomeruli of the kidneys combined. The normal adult GFR is 120 to 125 milliliters per minute, or about 180 liters per day. Maintenance of a relatively constant glomerular filtration rate is important because reabsorption of water and solutes depends on how quickly the filtrate flows through the tubules. It is held relatively constant, precisely controlled or self-regulated through intrinsic auto-regulatory mechanisms and extrinsic hormonal and neural mechanisms. If it is too high, fluid flows through the renal tubules too rapidly for them to reabsorb, and the required quantity of water and solutes is not properly reabsorbed. If it is too low, fluid flows too slowly through the tubules and waste that should be eliminated are reabsorbed into the bloodstream. The GFR is thus regulated by the phenomenon of renal autoregulation, which is the ability of the nephrons to adjust their own blood flow. It allows them to maintain a relatively stable glomerular filtration rate in spite of changes in arterial blood pressure. The next term we should understand is filtration fraction. Filtration fraction is that portion of the renal plasma which becomes the filtrate. It is the ratio between the renal plasma flow and glomerular filtration rate and is expressed in percentage. Filtration fraction is equal to GFR into 100, that is 125 ml per minute into 100, which is equal to 19.2%. Renal plasma flow is 650 milliliters per minute. The normal filtration fraction varies from 15 to 20%. The next factors are the filtration pressures. The pressures which determine the GFR are glomerular capillary pressure, colloidal osmotic pressure in the glomeruli, and hydrostatic pressure in the Bowman's capsule. The glomerular capillary pressure is the pressure exerted by the blood in the glomerular capillaries. It is about 60 millimeters of mercury and varies between 45 and 70 millimeters. It is the highest capillary pressure in the body and favors glomerular filtration. The next is colloidal osmotic pressure, which is the pressure exerted by the plasma proteins in the glomeruli. It is about 25 millimeters of mercury and opposes glomerular filtration. The third important pressure is the pressure called the hydrostatic pressure. This is the pressure exerted by the filtrate in the Bowman's capsule during filtration, and it is about 15 millimeters of mercury and opposes glomerular filtration. So, the net filtration pressure is the pressure between the pressures favoring filtration and the pressures opposing it. It is the effective filtration pressure or essential filtration pressure which is needed for the maintenance of GFR. Thus, net filtration pressure is equal to glomerular capillary pressure minus of colloidal osmotic pressure plus hydrostatic pressure in Bowman's capsule that is equal to 60 minus of 25 plus 15, which is equal to 20 millimeters of mercury. The normal value of effective or net filtration pressure is 20 millimeters of mercury and ranges between 15 and 20 millimeters of mercury. Now, we move on to the stage 2 of urine formation, namely tubular reabsorption and secretion. The second stage of urine formation is tubular reabsorption and secretion. This involves removal and addition of chemicals after the glomerular filtrate leaves the Bowman's capsule and enters the renal tubule. The renal tubule is very long, which increases its absorptive surface area. It reabsorbs about 65% of the glomerular filtrate while it removes some substances from the blood. Tubular reabsorption is the process of reclaiming water and other substances from the tubular fluid. Tubular reabsorption begins as soon as the filtrate enters the proximal convoluted tubule and involves near total reabsorption of organic nutrients and the hormonally regulated reabsorption of water and ions. The most abundant cation of the filtrate is sodium, and reabsorption is always active. There are two types of reabsorption processes: passive and obligatory processes. Passive tubular reabsorption is the passive reabsorption of negatively charged ions that travel along an electrical gradient created by the active reabsorption of sodium. Obligatory water reabsorption occurs in water permeable regions of the tubules in response to the osmotic gradients created by active transport of sodium. Absorption of glucose, amino acids, vitamins and most cations occurs when solutes are transported with the sodium when it moves along its concentration gradient. This method is called secondary active transport. Substances that are not reabsorbed or incompletely reabsorbed remain in the filtrate due to a lack of carrier molecules, lipid insolubility or large size. Different areas of the tubules have different absorptive capabilities. The proximal convoluted tubule is the most active in reabsorption, with most selective reabsorption occurring here. The descending loop of Henle is permeable to water, while the ascending limb is impermeable to water but permeable to electrolytes. The distal convoluted tubule and collecting ducts have sodium and water permeability regulated by the hormones aldosterone, antidiuretic hormone and atrial natriuretic peptide. After water and other substances leave the surface of the renal tubule, they are reabsorbed by the capillaries into the bloodstream. Tubular secretion is the process in which the renal tubule extracts chemicals from the capillary blood and it secretes them into the tubular fluid. The tubular secretion disposes of unwanted solutes, eliminates solutes that were reabsorbed, rids the body of excess potassium and thus controls the blood pH. Tubular secretion is most active in the proximal convoluted tubule but occurs in the collecting ducts and distal convoluted tubules as well. This process serves two main purposes. One is waste removal. Urea, uric acid, bile acids, ammonia and creatinine are secreted into the renal tubule. Tubular secretion clears the blood of pollutants and drugs as well. One reason why so many drugs must be taken 3 to 4 times per day is to maintain the therapeutically effective drug concentration in the blood to compensate for the rate of clearance through tubular secretion. Maintaining the acid-base balance. Tubular secretion of hydrogen and bicarbonate ions serves to regulate the pH of the body's fluids. Below you can see the different stages and sites of tubular reabsorption. The first two columns give us the part of the nephron or the site at which the reabsorption takes place. The second column gives us the different kinds of substances which are reabsorbed, and the third column shows the mechanism by which the reabsorption takes place. In the first or the proximal convoluted tubule, you can see that the ions that are reabsorbed are sodium ions. Virtually all nutrients like glucose, amino acids, vitamins, and some ions like chloride, potassium, bivalent calcium, magnesium are reabsorbed. Bicarbonate is reabsorbed. Water is also reabsorbed. Lipid soluble solutes and urea are reabsorbed. The mechanism is by primary active transport, basolateral sodium and potassium pumps. The secondary active transport with sodium, passive paracellular diffusion driven by electrochemical gradient, secondary active transport linked to hydrogen ion secretion and sodium reabsorption, osmosis driven by solute reabsorption or obligatory water reabsorption, passive diffusion driven by concentration gradient created by the reabsorption of water, and primarily passive paracellular diffusion driven by chemical gradient. In the nephron loop, that is in the descending and ascending limbs, the reabsorption of water, sodium, chloride and potassium, calcium and magnesium ions takes place through osmosis, secondary active transport of the ions, and passive paracellular diffusion driven by electrochemical gradient. In the next region, that is the distal convoluted tubule, sodium and potassium ions and calcium ions are reabsorbed by primary active sodium transport and passive uptake modulated channels in apical membrane, primary and secondary active transport in the basolateral membrane. In the next region, namely the collecting duct, sodium, potassium, bicarbonate, chloride, water and urea are reabsorbed by active transport, paracellular diffusion, osmosis and facilitated diffusion in response to concentration gradients. Next, we move on to the regulation of urine concentration and volume. One of the critical functions of the kidney is to keep the solute load of body fluids constant by regulating urine concentration and volume. This function is carried out through the counter current mechanism, which traditionally involves the counter current multiplier and the counter current exchanger. The first takes place in the region of the nephron called the Henle's loop, and the second occurs in the region of the peritubular capillary bed called the vasa recta. Both are involved in establishing an osmotic gradient throughout this region. Counter currents exist when fluids flow in opposite directions in parallel and adjacent tubes. The two limbs of Henle's loop are a counter current. The two limbs of the vasa recta are also counter current. Thus, it is apparent that these two sets of tubes are parallel and adjacent, as you can observe in the figure. Next, we move on to the Henle's loop. The portion of the nephron called the Henle's loop is shown in the right half of the illustration. It consists of a descending limb which has a down arrow in it and an ascending limb with an up arrow in it. The ascending limb has a thick and a thin segment. The dots which represent the various solutes that become more concentrated in both limbs towards the bottom of the loop. The thick walls of the ascending limb indicate that this region is impermeable to water. In the vasa recta, the capillary bed is shown in the red color and at the left of the illustration. It also consists of an ascending limb and a descending limb identified by the arrows in each of the areas. Again, the dots represent various solutes that are more concentrated at the bottom of the loop. Here, examination of both limbs of the vasa recta shows the concentration of solutes by the number of dots, and it is the same at any horizontal level. However, when we imagine the fluid flowing through the vasa recta for a short distance and then stopping, we compare the concentration of the solutes at any level, they will not be the same. At any level, the solute concentration in the descending limb will be less than in the ascending limb. But because both limbs are freely permeable, sodium chloride diffuses from the ascending into the descending, while water diffuses from the descending to the ascending limb, as shown by the block arrows. When equilibrium is reached, both limbs once again have the same concentration of water and solutes. Water is thus exchanged for sodium chloride, and this facilitates the counter current exchange mechanism. Next, we move on to the counter current multiplier mechanism. The above described counter current exchanger would not exist if there were not some mechanism to initially make the vasorecta more concentrated at the bottom of the loop. This is accomplished by the loop of Henle. The ascending limb of Henle at the early distal tubule is impermeable to water, as indicated by the thick wall. These regions actively transport sodium chloride out of the filtrate and into the surroundings. The asterisk in the block arrow indicates that a more complex mechanism is involved, but the net effect is that only sodium chloride is moved out of the filtrate. The sodium chloride diffuses into the descending limb of the vasorecta. Any that might diffuse into the descending limb of Henle will only get pumped back when it enters the ascending limb. So, this is not shown here in the diagram. It will not diffuse into the ascending vasorecta because the fluid is already highly concentrated. This is the mechanism that multiplies the concentration of sodium chloride in the descending vasorecta, making the counter current exchange possible. The next phenomenon is osmosis. Both blood and filtrate descending into their respective loops have low solute concentrations. Both flow beside upcoming columns having high solute concentrations. As they move past one another, water from the downflowing fluid columns will diffuse into the more concentrated upflowing columns. However, only the upflowing vasorecta is permeable to water, that is, it is returned to blood and none to the filtrate. This also ensures that high solute concentration will be maintained at the bottom of both loops. Next, coming to the excretory products, urea diffuses passively among and between the lower portions of both the blood and filtrate, as indicated by the block arrows. The result is that half the urea is excreted and half is kept in the body. In the figure, you can see that the concentration and osmolality of the fluids in the loop of Henle and vasorecta. The filtrate enters the descending limb and becomes progressively more concentrated as it loses water. As indicated in the pink columns, blood in the vasorecta removes water, leaving the loop of Henle. And finally, the ascending limb pumps out sodium, potassium and chloride, and the filtrate becomes hypoosmotic. Next, we come to the concentration and volume of urine. The three key players in urine formation are the counter current multipliers, the counter current exchangers and the collecting ducts. In addition, there is the role of the juxtaglomerular or juxtamedullary nephrons. Here you can see the juxtamedullary nephrons and their role in the process of urine formation. Since water is freely absorbed from the descending limb of the loop of Henle, the filtrate concentration increases and water is reabsorbed. The ascending limb is permeable to solutes but not to water. The effect of reabsorption and concentration is depicted in the figure. The vasorecta aids in maintaining the steep concentration gradient of the medulla by cycling salts into the blood as it descends into the medulla and again as it ascends towards the outer cortex. The vasorecta thus helps to preserve the concentration gradient. The entire length of the vasorecta is highly permeable to water and solutes, but due to the counter current exchanges between each section of the vasorecta and its surrounding interstitial fluid, the blood within the vasorecta remains nearly isoosmotic to the surrounding fluid. As a result, the vasa recta do not undo the osmotic gradient as they remove the reabsorbed water and solute. At the collecting ducts, under the control of the antidiuretic hormone, the collecting ducts determine the final concentration and volume of urine. In the collecting duct, urea diffuses into the deep medullary tissue, contributing to the increasing osmotic gradient encountered by the filtrate as it moves through the loop. Since tubular filtrate is diluted as it travels through the ascending limb of the loop of Henle, production of a dilute urine is accomplished by simply allowing the filtrate to pass on to the renal pelvis. Formation of concentrated urine occurs in response to the release of the antidiuretic hormone, which makes the collecting ducts permeable to water and increases water from uptake from the urine. Here you can see a summarized events occurring in the renal tubule and in the peritubular capillaries. So the blood flows into the glomerulus through the renal artery, and it is filtered at the region of the glomerulus and the Bowman's capsule. The glomerular filtrate enters the Bowman's capsule, and here you can see that there is an exchange of ions between the peritubular capillaries and the renal tubule. Sodium ions are reabsorbed by active transport. Chloride ions, phosphate bicarbonate are negatively charged ions are attracted to positively charged sodium ions, and they are removed by passive transport. As concentration of ions increases in plasma, the osmotic pressure increases and water moves from renal tubule to capillary by osmosis. Sodium ions are reabsorbed by active transport and negatively charged ions by passive transport. Thus, the concentration of solute increases in plasma and osmotic pressure also increases. To compensate the concentration, water moves from the renal tubule by reabsorption. The fluid then moves into the collecting tubule and is propelled towards the urinary bladder as urine. Next, we move on to the ureters, urinary bladder and the urethra. Freshly voided urine is clear and pale to deep yellow in color due to urochrome, a pigment resulting from the destruction of hemoglobin. It is slightly aromatic but develops an ammonia odor if allowed to stand due to bacterial metabolism of urea. It is usually slightly acidic. The pH is around 6 but can vary from about 4.5 to 8 in response to changes in metabolism or diet. It has a higher specific gravity than water due to the presence of solutes. Urine is about 95% water and 5% solutes, the largest solute fraction devoted to nitrogenous base, namely urea, creatinine and uric acid. Human urine consists primarily of water with organic solutes, including urea, creatinine, uric acid and trace amounts of enzymes, carbohydrates, hormones, fatty acids, pigments and mucins, and inorganic ions such as sodium, potassium, chloride, magnesium, calcium, ammonium, sulfates and phosphates. The chemical composition of urine is as follows: water 95%, urea 9.3 grams per liter, chloride 1.87 grams per liter, sodium 1.17 grams per liter, potassium 0.75 grams per liter, creatinine 0.67 grams per liter, with lesser amounts of other ions and compounds. The ureters are tubes that actively convey urine from the kidney to the bladder. The walls of the ureters consist of an inner mucosa continuous with the kidney pelvis and the bladder, a double-layered muscularis and a connective tissue adventitia covering the external surface. The urinary bladder is a muscular sac that expands as urine is produced by the kidneys to allow storage of urine until voiding is convenient. The wall of the bladder has 3 layers: an outer adventitia, a middle layer of detrusor muscle, and an inner mucosa that is highly folded to allow distension of the bladder without a large increase in the internal pressure. The urethra is a muscular tube that drains urine from the body. It is 3 to 4 centimeters long in females but closer to 20 centimeters in males. There are two sphincter muscles associated with the urethra: the internal urethral sphincter, which is involuntarily and formed from detrusor muscle, and the external urethral sphincter, which is voluntary and formed by the skeletal muscle at the urogenital diaphragm. The external urethral orifice lies between the clitoris and vaginal opening in females, or occurs at the tip of the penis in males. To conclude, urine formation is a complex process by which metabolic wastes and water are removed from blood. It is carried out by the kidneys, which contain the functional units called nephrons. Each nephron is constituted by the glomerulus, the renal tubule, which drain into the collecting tubules. It involves three major processes, namely glomerular filtration, tubular reabsorption, and secretion and concentration of urine. The urine formed is drained into the urinary bladder through the ureters and is expelled out through the urethra.